3 weeks. So, dear participants, you have been there are nearly 250 participants in this workshop, and it's a wonderful achievement by NPT and spearheaded by Dr. Ganesh Dakle and his team from AIMS Nagpur. And in the last 3 weeks, you have seen you have been taken step by step on how to implement good clinical practice in your clinical trials and day-to-day practice of clinical research. And today you will be seeing the most important document of them all, that is ICH GCP guidelines, the revised guidelines R3 2025 version. And Dr. Ganesh, kudos to you for getting across so many participants and so many speakers, eminent speakers, not only from INIs, but also from various other peripheral medical colleges in India, and for giving them a chance and coordinating this wonderful mammoth event across 4 weeks. Best of luck to all the participants, and hope you had a wonderful learning session in the last 3 weeks. Thank you, Dr. Ganesh. Thank you, NPT. Over to you, Dr. Ganesh.
Thank you so much, sir, for your concluding words for the workshop as a chairperson of the GCP workshop 2026. So today's first topic is on silent features of ICH GCP E6 R3 2025, which will be taken by Dr. Nusrat Shafiq Madam. And the second session is on handling protocol deviation and violations, which will be taken by Dr. Santosh Taur sir. So the first session will be moderated by Dr. Shashidhar Bannale sir. Sir is presently working as Professor Pharmacology, MRMC, Kalaburgi, Karnataka. Sir is also having experience of 18 years of undergraduate teaching as well as 15 years of PG teaching. Sir's achievement is Sir was gold medalist in MD for RGUHS University, Bangalore. Sir is also acting as a IAC chairman at BLD University, Bijapur. He has also been involved in international multicenter clinical trials involving women and children's health research. In the name of the sir, there are 16 international publications and contributed to 2 textbooks of pharmacology. And sir's interest is in maternal and child health, medical education. So I welcome you, sir, for the session. The second session, which will be moderated by my friend Dr. Sarfaraj Khan. Sir is presently working as professor and head pharmacology, Chhindwara Institute of Medical Sciences, Chhindwara. Sir has worked as medical advisor in pharma industry for 3 years. He also acted as the resource faculty for National Workshop on GCP and Bioethics. He is also a life member of NPT as well as ISRPT, and he is also the member of editorial board of Journal of Contemporary Medicine and Dentistry. Sir is also having several publications in national as well as international journals, and he is also a member of IEC, CIMS Chhindwara as well as member secretary of Pharmacovigilance Committee. I also welcome Dr. Sarfaraz sir as a moderator. Now I request Dr. Bannale sir to introduce the first speaker, Dr. Shafiq Madam. Over to Dr. Bannale sir.
Thank you, sir. At the outset, I would like to thank our Ganesh Dakle sir, President of the NPT, and Dr. Kamal sir, Coordinator, for this wonderful opportunity to moderate the session. Thank you once again for the kind introduction. I would like to introduce our first speaker today, Madam Dr. Professor Nusrat Shafiq Madam. She is a clinical pharmacology. She is from the clinical pharmacology unit, Department of Pharmacology, PGI Chandigarh. She is also, Madam is also member secretary of IEC for drug trials from PGI Chandigarh. Also the co-chair of IEC AIMS Mohali, ex-president of Society of Antimicrobial Practices of India. Also an expert consultant for the National Action Plan AMR2, also the review committee for novel antimicrobials with the WHO. So, Madam is also PI for India Hub for Center of Antimicrobial Optimization Network Lead PGIMR, that is ICMR Phase 1 network. And she has many publications to her series. Now, this is a short, very brief introduction of the Madam. Now, I would like to take the Madam to the, welcome to the Madam to the stage, and let the Madam go to the topic. That is changes in the E3 guidelines as compared to the older version?
Thank you. Thank you, sir, for that kind introduction. At the outset, I would like to thank the organizers and respected chairperson for setting the stage for this session. Before I go into my presentation, I would really want to echo what Dr. Balakrishnan mentioned in the beginning. It is a mammoth exercise that has been taken by NPT, and it is turned out so beautifully and smoothly. Was listening to some of the recorded presentations, and I must say, I do not think I will be able to match up to such erudite speakers who preceded me, but let's see. So having said that, thank you once again, and we start with the presentation, which is about the third version of ICH GCP, which is recently we all are updating ourselves about the same. Please bear with me because the slide shifting is taking time. Yeah. So I'll just give you a brief overview of how I planned the presentation. You all have learned quite in details about GCP, but there are certain aspects which I think I'd like to touch upon once again. Then I'll talk about just the key domains of changes in ICH E6 R3, especially the quality by design and proportionality principle. It is for everyone to go and read it word by word, line by line, and I think I will not spend too much of time there. Some key differences from R2 or the preceding version. We'll touch upon that and spend some time on data governance. And then we'll see how we need to pull up our socks in accordance with ICH E6R3. So that's a brief outline, but I have a tendency to deviate from what I propose, so please bear with me. And then I'll conclude by summing up the 11 principles of GCP in the third revision. So before I go into the topic proper, I would like to just make you think a little because, and when I say you, it is for the young initiators who are just walking into this area of clinical trials and learning about various aspects. So you've heard in the previous presentations about all the tenets, principles, and everything else. But there's something which happened in the background when the ICH-GCP guidelines were floated for the first time, and more importantly, when they came up for the second version came up, a lot of regulatory updates had to be taken up to match up with the requirements of GCP. And our own regulatory body, that is DCGI, is the CDSCO, did also went ahead and formulated certain guidelines, came up with the Schedule Y and subsequently NDCT 2019. Simultaneously, pharmaceutical companies had to reorganize their work, especially those who were into clinical trials space. They had to reorient and reorganize themselves to a great extent. We all know that there was a mushrooming of contract research organizations because the operations had to be done so methodically and so carefully that the sponsors thought that it is best to delegate the responsibilities to people who would have local branches there and carry out day-to-day operations. This was also associated with the innumerable training programs. I remember way back in 2001 where in our department had organized one such workshop along with industry partners. But after that, we've had a series of such programs being undertaken all over the country. And importantly, a whole new workforce was generated. We know clinical research associates, clinical research coordinators, many other data entry operators, and there was an industry, virtually an industry came up just because of a simple document, ICH GCP. The aim, we all know very well, was about quality of data, ensuring rights and safety of participants, and assurance in results. So that was the large aim, which was usually voiced in all the GCP training processes. But we also know that this excessive documentation, which went on to coining of an adage wherein it became GCP is equivalent to documentation, also came up at this time, and which also, I would say, led us to get a little more focused on documentation. Things about patient safety somehow took a backseat. Not exactly, but that this primary focus should always have been patient safety. But in my experience, I've seen that in the process that that became a little concerning. Then there was a diminished space for small industries. Industries, small and medium-sized industries, especially for countries like ours, wherein people were just trying to get their space in R&D, especially clinical trials, conducting clinical trials for their own products. They found it a little intimidating to match up to the processes. And then the cost of conducting such GCP compliant trials became exorbitant. It was beyond reach of many of these industries to undertake such studies. But having said all that, we all can very well appreciate, yes, the quality of data did improve, rights and safety of participants did get attention in many other ways, and we had more assurance in results. We all know that the current version was finalized in 2025, but before this, we had some of these elements taken up in the 2016 versions as well. And the foundation, we know, that was laid in the first version, which came up way back in 1996. And the foundation stone of all this was Declaration of Helsinki. I'll not go into those details because they have been dealt with in the previous session. So for this version, I think I'll focus on some of the important, very key things, which are terminologies and concepts which have been brought in here. And these would be as detailed out in this slide: quality by design, proportionality principle, integration with technology, and adaptation to new clinical trial designs. So these are some of the aspects that I'll touch upon. I'll waver a little from this broad thing. So let's just talk about quality by design approach. What do we mean? In fact, it took me some time to decipher it and to do a lot of reading. And I thought, let me see if it is really something very, very new, or it's something which was there and has been repackaged, which is often the case. So whenever you go back to the previous versions, you would have heard about the terms quality assurance and quality control. Now, some of you who are relatively new in this area, quality assurance refers to all the proactive preventive measures that you take in order to ensure the data quality, the quality of ensuring the safety rights and all those things. So that is a proactive preventive kind of approach, which is done before you start with the trial. And these would entail things like making the SOPs, providing the right trainings, getting all your documents properly, laying down the procedures. All those things would come in quality assurance. While quality control is more like a reactive approach, wherein you ensure some kind of activities like monitoring, wherein you see how while the trial is progressing, if there is a compliance to the protocol, if all the other aspects of the trial which relate to ethics are being taken care of, if the data integrity is ensured, and all these things are taken care of. Now, so that means that this was always a part of any GCP document that we have had so far. So what is this quality by design? So well, a brief understanding about this concept is important. I think it was Joseph M. Juran which first floated it in one of his seminal works, wherein he talked it in, he first mentioned it in the context of drug development, about a product development, wherein there was a concept that you lay emphasis on testing approach. From that, he said we should lay down systems and processes in place which ensure certain level of quality just because the processes have been so thorough. So that concept was Sorry, are you saying something, Dr. Dakle?
You can go ahead, madam. You can.
Yeah, sorry. So, so that concept was adopted into clinical trials, and if you think about it, it is all about ensuring
Shilpa,
ensuring
just a minute. Shilpa, you can do one thing. You
Shilpa,
can
you can mute
all the
the
participants.
background noise.
ensuring, so thank you, ensuring all the processes which will prevent any kind of compromise in quality of the clinical trials. So this has to be done for designing the protocol and in the process of operationalization. Now that means what is the purpose? So would think that it will be reflected only in the monitoring protocols? No. So herein lies the importance. It starts figuring in at the stage of protocol writing. We'll go into some details of the same. It is encompassed in the entire process of analyzing the results and also, of course, in the management plan. Importantly, now the focus is on identifying some of the parts of the aspects of the clinical trial which will be considered as critical to the quality. For example, I can say randomization is critical, or masking is very critical, or retention of patients is very critical for my trial. So those would be identified in advance, and strategies and actions for efficiently supporting quality in these critical areas will be ensured. Not to say that other things will not be kept in mind, but these will be under the focus of attention. From a concept of total absence of errors, which was actually very impractical, it has moved on to absence of error that matters. In fact, most of us who were doing academic clinical trials, multicentric academic clinical trials, we always had this idea at the back of our mind and we would talk about it. It is just that we did not package it into its terminology and present it forward. So, but it also involves certain other things which have to be carried out. Most importantly, for undertaking such an exercise, now it doesn't remain a checklist kind of thing any longer. You have to involve stakeholders. And what are the important stakeholders that you have to involve? Investigators, maybe not all the investigators that are going to be part of the trial, but investigators who are aware of the practice setting, investigators who are aware of the trial related issues, and they have to be roped in right at the beginning for for planning it out. Then not only investigators, but patient advocacy organizations called POW, acronymed POW, have also been paid attention to in the R3 version. One of the important reasons I see, though it is not mentioned like this, is because they're talking increasingly about decentralized trials, and therein having inputs from the patients would be very important in planning out the entire study. Now, to make it a little more clear, I was wondering, how do you actually operationalize on the ground? How does it translate to your quality of design is a very big term. So I went to the site, which is the CTTI. It is it's a Clinical Trials Transformation Initiative. It is a group which is formed under USFDA, which partners with Duke University, and they have done a lot of work on quality of design. They have created tools, they have created documents, guidelines. I would recommend that everyone goes through it, and this is the example is taken from there itself. I read through it and I had to do a lot of back and forth reading. I've restructured it a little. And what I realized is I had presented to you this way. Whatever we talked about, critical to a clinical trial would be the concept of randomization. Because it is key to avoiding selection bias, and then it is the bedrock for proper comparative statistics or inferential statistics that have to be drawn out of the trial. So it's very important that randomization is identified as, for a given trial, as a critical to quality parameter. Then what you do is you do a brainstorm. something like that, what you do in your critical evaluation of literature. So on the left side, you would see certain elements that I have written down, and these are the points that you actually discuss in general club. So you would discuss, were there any potential for prediction of treatment allocation? Was it possible to predict the treatment allocation? So obviously, were block sizes allowed? Did they allow it that way? Was the algorithm such that it could have led to dilution of randomization? Was there any way that allocation concealment was not put in place, or participants' condition could have come in way of randomization? Or during the conduct of study, how would it be ensured that the trial participants actually received what they were randomized to? And if for treatment of withdrawal, what would be done if the patient is withdrawn before to treatment administration? What was done in a study? So these are the things we would take into consideration while planning out the randomization component of our study protocol. Then what we do is, in order to evaluate a protocol on this, we will define certain parameters. Was the study randomized? Yes, if it was. Who will generate and implement randomization? How will it be carried out? Are approvals needed? For any case, there are certain situations where approvals may be needed before you actually recruit the patient or you proceed with randomization. Who will execute it at trial site, and how and by whom will randomization errors be managed? So that makes the entire pathway for quality of design process for just one aspect, which was identified as a critical aspect. I will not go into the masking in the interest of times. You can maybe take it as an exercise and perhaps see how the same principles could be applied to masking. So at this point, I want to say that it is a process. Quality by design is a process, and it starts at the time of draft study concept. Some of you who would have attended our national workshop of clinical pharmacology, we emphasize a lot on writing the draft concept. And here itself, they say that you should identify the critical factors, identify what could be the risk, and I mentioned some mitigation steps. For example, if we identify retention of trial participants to be critical quality factor, then risk would be, if in the first visit, say if the patient is spending some 6 to 8 hours, that would be an important risk factor which can affect this critical component. Then we have to think about what steps can we take to reduce this duration in order to retain the patient. So that becomes your first draft. You improvise upon it through different steps. As you move along different steps of protocol development, even a protocol matures. And then during the conduct of trial, you have to have a repeat evaluation of these parameters and update. This may even warrant protocol amendments. So that has to be taken care of. And when the study closes out, you don't stop it at that point. You reflect what were the things that worked for me and what did not work for us. And then that can be applied in the subsequent principles, subsequent protocols that any team develops. There is also a concept of quality tolerance limits, which is not mandatory. For some of the aspects, we can have tolerance limits. Beyond this, we will be really alarmed. Say, for example, if the attrition rate goes beyond 15%, that is our quality tolerance limits, and it should get everyone on board to rethink about the mitigation strategies. Then we come to, so that was about quality of design principle. I hope I've been able to make it a little clear to myself and to everyone else. But if not, I'll be happy to take it in more details later. Then we come to the concept of proportionality principle. If you read it very carefully, it is about parsimony principle, because clinical trials were becoming, the processes were becoming so burdensome. They thought that, okay, we will make the processes more elaborate and burdensome only if the process is related to patient safety, or if it is very critical to the data or the trial results. Say, for example, if it is endpoint assessment, they said herein will be our focus. If it is, say, for example, sorry. Then, say, for example, if it is a trial which involves mortality as an important endpoint assessment, then, and it is not an infection-related trial, then everything else will not be paid too much of attention. There is a catch there. This site gave the example of CTVS trial and cardiac endpoints as the primary endpoints, and they said temperature may not be important, but we must not remove our eyes from that, because it could be very important, because it could be an indication for any impending infection and would be critical to patient safety. So basically, herein they say that the amount of effort that is put into any trial related step should take into consideration risk-based approach. It should be fit for the purpose, just appropriate for the purpose. It should consider the relevance of the data, and it should all be done in a proactive manner. How does it impact? It impacts monitoring, most importantly. From every trial requiring detailed on-site monitoring, there is a possibility of making it very risk-based kind of monitoring, very or decentralized monitoring. Then emphasis that has to be placed on documentation at various levels, that gets modified. You make it proportional to what is critical. And if there are computerized systems, they would require very thorough system validation, no doubt. But it has to be proportionate to the requirement of any particular aspect. So that is what is the principle 7, or the principle of proportionality. All in all, it is about flexibility. So that was about principle of proportionality. Then we just touch upon certain other differences which are there from R2. I think there is one we should be thinking about. That is, now it is not mandatory to report all SUSARs suspected and expected serious adverse drug reactions in an expedited manner, and they propose an alternative arrangement. Here, I think it is better we go a little slow on this, and alternative arrangements for following our SAE regulations. We do have alternative arrangements for this already in place. There are sections which relate to upgrading process for novel trial designs, especially those trial designs which involve decentralized trials wherein the data collected right from the patients and the study material is supplied to the patients directly, or it is real world designs. In fact, there is an Annex 2 which is going to come up very shortly and notified shortly, which relates especially to special considerations. In these, it has not been notified. I have not gone into the details, but important elements there are confidentiality. How do you manage side effects in such situations? So those are the things I think I will be open to discuss a little more. Things about consent in such trials. These are some things which need to be looked up to differently. Now, as I mentioned earlier, there is a concept of involving the people who are going to be part of the study. Not everybody can be involved, or some representative groups can be included. Participant diversity is something which is emphasized upon. In fact, it is said that if you exclude a participant, it should be explained why a patient participant has been excluded. So we can understand from this that the emphasis is for the generalizability of the results. So striking, earlier it was totally on the internal validity. Now they're opening up to external validity. An important section has been added, that is the data governance section. And one section on non-therapeutic trials has been removed. So data governance has got a special emphasis now. There is a dedicated section wherein it has been emphasized that the data should be the integrity, traceability, and security of data must be ensured, so that there is accurate reporting. It can be verified at any time, and interpretation is based on the actual data. Especially when you use computerized systems, trial participants' confidential data has to be really taken care of, especially if it is if the data is picked up from electronic health record systems, etc., or it is picked up from digital health technologies. Management of computerized systems for ensuring that they're fit for purpose has to be taken care of. Elements of clinical trial, essential elements of clinical trials should be safeguarded to the last minute, and important key decision-making processes should be which are around the data should be taken care of. And these have to be taken care of from the time of data capture to the time of data finalization. So they say that all this, at every step, there has to be a plan, not just about the raw data as it gets captured, even the relevant metadata about all the logs, everything, that also has to be very properly maintained. There should be a proper audit trail. This should be reviewed from time to time, and any corrective actions that are needed should be taken in a very reasonable time. Care has to be taken while transferring data from one system to another system and when finalizing the data. Importantly, we have to understand that now, as we're moving to digital technologies, access to user, denying access after a certain period of time, contingency arrangements for system failures, all these have to be taken care of in any trials that we do using these systems. So I'll just come to this aspect. How do you upgrade Indian regulations? First question is we have to ask is, do we need to? To some extent, we need to. But who will be the stakeholders? We have to draw out of people who are conducting such trials, who have a good understanding, industry partners, CROs, all those people will be important. Decentralized trials, I think we need a dedicated section on that. I have myself experienced the issues with consent and written about it, and I think we need to see it in the light of Indian population and our kind of patients that we meet. Data governance systems are easy said and then done, but in the process, if things improve, it should be deliberated upon. If not in totality, we can go in a stepwise manner. Remote monitoring is already happening, and validation of remote instruments, I mean, digital health technology is something that I think needs a very deep emphasis, because we always raise this issue that where will the data be captured? Who will have an access to this data, which is generated out of digital watches and all those things? Unfortunately, our electronic health record systems are not enabled to allow clinical trials till now, to allow clinical trials to access this data from there, but we have to gear up for such a situation. And then we have to brace up to risk-based monitoring. They advocate discretion in reporting SUSAs. I think there we can follow our own principles. I think we will be better off with that. That's my idea, but I think a consortium kind of approach will be better. The people will have different approaches of looking at it. So that's all, but I would like to end up by summarizing it all into the 11 principles of E6 R3. From 13, they have become 11. So basically, we'll just briefly go over it. They're still grounded in the Declaration of Helsinki. Informed consent has got a separate and a very detailed emphasis, and it is said that assent forms must be reviewed wherever applicable. And there are more nuances to it, which I think I can take up in discussion. They say that EC and IRB should be independently reviewing, but important difference is that it can be, they mention explicitly that it can be one body which does it. All the scientific exploration should be based on best current knowledge. There should be scientific soundness in any trial. and this should be updated as the knowledge advances. Qualified individuals should undertake study. And as far as our regulation about a physician undertaking a clinical trial as a principal investigator, ICH GCP does not advocate says. It says that they should be part of the team in order to take care of the medical requirements. Quality in design and operation and proportionality principle we have already touched upon. The protocol should be clear, concise, and scientifically sound, which is based on the previous two principles. It should lead to generation of reliable results. Importantly, documentation of roles and responsibilities should be very explicit at each level. Sponsors, when they delegate it to CROs, that also should be clear. Importantly, investigators now have a right to say whether they want to take services for any of the adjuvant activities related to clinical trials, for example, for an NABH accredited lab evaluation or for image reading protocols. Quality of clinical trial product and its management should be as per the GMP requirements for a given state and as required by the regulators. So all in all, preventive approach is something which has been emphasized upon. Thoroughness is emphasized, but it should not be burdensome. Robust digital frameworks are something which have been, which are now to be taken care of. And there should be an eye on data through its life cycle in order to ensure ethical and scientific conduct of trial and generate reliable data. These are some of the reading materials, not material, but links, which I think would be very useful and informative. And I think I'll be happy to take any questions, and I hope I was in time. Thank you very much for patient listening. Thank you. Shall I
Madam,
stop
just
sharing?
Yes.
Yeah. Yeah. So if there are any questions in the chat and these can be more taken up, I'll be happy to take any answers.
Thank you, madam, for a very comprehensive talk on the recent changes in the R3 guidelines.
Thank you, sir.
Madam, there is one question in the chat box. Can you elaborate something on fit for purpose in detail in clinical trial?
Yeah, so let us just take the example of it is not at one level, it is at a different levels. Say, for example, it is about data capturing aspect. And if I have to take a data on major adverse cardiac events, for example, for me, what is important? Repeat angiography, myocardial infarction, cardiovascular death, or death. Anything which is happening in between, for example, patient's temperature, patient's body mass index may be important, but say patient's bone mineral density and patient's surgical complication, for adverse event, it may be important. For efficacy, it is not important. So when I generate my systems for capturing this information, I have to fit it for the purpose of the efficacy. That is one way in fit for purpose can be looked at. The second aspect is, for example, computerized systems validation I have to look into. Now, for something which is late phase trial, I may not require very extensive validation processes, which itself would require an industry to carry it out. But key critical aspects I can outline and say I will ensure that the access to computer systems for entering data is restricted. It is the access is changed as per procedure when required. I can ensure that metadata can be analyzed on a timely manner. I can ensure that the transfers, when they happen from one system to another system, that is taken care of. I needn't have things like I'll ensure that some kind of viruses don't and corrupt that system. But I really do not have to go into very intensive security systems for this kind of study. So that is, in essence, what is fit for purpose. Every step has to be as required. It doesn't have to be deep, very, very deep. For example, if it is monitoring, if the purpose of monitoring is to find the reporting of adverse events which has been done in a timely manner, then I'll focus to just that. Then I can even think that maybe I can have my monitoring mechanisms totally decentralized. If it's a phase 4 study, there can be somebody who will be sitting at the other end, looking at the data at regular interval, and say that monitoring will be done if, say, for example, I see that the quality threshold limit has crossed my pre-specified limit, then we will have an on-site visit. So that is the plan that you have, which is fit for the purpose of the monitoring of a given trial. So fit for purpose is a broad concept which is applied to different aspects. In the document, emphasis has been laid on to the computerization, digital systems which are involved in trials, but I think it is also distributed, but you have to read it between the lines. So I hope I was able to answer your question.
Yes, yes. Madam, second question is how quality by design is taken care of in decentralized clinical trials?
So again, when we say decentralized clinical trials, it will have to have different critical components identified, critical to the trial. So one of the components that I would identify is the consent, because it is just one of the things. The other things can be critical to quality, would be retention of these patients, supply of the trial material to them, identification of adverse events in a timely manner. These could be many others, but let's just take the examples of identification of consent. Now that we have said what could be the risks with taking the consent, if I imagine our own population, the risk that I understand is non-familiarity with technology. Second thing is somebody else giving consent on someone else's behalf, especially if it's a woman participant. Husbands would be taking the call. So that kind of risk I have to identify in the context in which we operate, or patients not comprehending all the aspects, but not being very comfortable with the participants. Sorry, it's not patient, participants not coming up with the queries. So those are the risks that I've identified. Then what will be the risk mitigation strategy? And a simple risk mitigation strategies, I build in a comprehension assessment system, or I can actually ask some questions, give them time to understand, have an identification document in between, have an video on while taking the consent. So this is at the time of the protocol. While doing the study, I may come up, identify other factors, and then address the same. So this was just one of the parameters that I have explained to you for a decentralized trial where the patient is sitting somewhere else and we are recruiting the patient and then subsequently following them up through the trial. There could be other aspects and similar principles could be applied there. If you wish to give me another example, I can take it up if the chair decides there is time for that. I hope it was clear.
Yes, madam. Yes. You
Yes,
can
ma'am.
go ahead
Thank
if you
you.
want to go ahead.
Yeah, so the other aspect could be something like, detection of adverse event. In fact, we faced this situation in our ethics committee. Was it our ethics committee or Ames-Woodpur? I don't remember, but one of the ethics committees wherein we had this question. Now, adverse event is occurring. All the lab reports are directly going to the sponsor. How will the investigator know? Here, there was an investigator who was in between and who would be watching the lab reports or would be privy to lab reports in timely manner. The patient may not actually perceive it as an adverse event. So I have identified, and how did it emerge? It emerged as a discussion with the stakeholder. You identify it as the critical factor would be timely detection of adverse event and its reporting and management. What are the risks? Risks are delayed detection simply because the information reaches late to the investigator. Mitigation strategy is a copy of the same has to be contemporaneously delivered to the investigator. So those are the risk mitigation strategies. You improve upon it when you do when you conduct the trial. You can come up with a better strategy and then you update it in the protocol. And at the end of the study, you may identify what worked, what did not work, and then apply it to the next trial. That would be another example. Why?
Banale sir, one more question in chat box. Can you go
Yes,
ahead
sir.
with that one?
Yes, what?
Can we use?
Can we use Aadhaar-based biometric face capture for consent? Madam, this is the question. Can we use Aadhaar-based?
So now it is very important that you have confidentiality of data and data security in place. If you can ensure that, I think we do it sometimes. In fact, for phase 1 studies, we do when we keep a copy of Aadhaar, but it has to be kept in a very secure manner, and we are not using digitalized system. The whole issue becomes difficult if digitalized systems are not properly secure. You can use, but you must identify the person. It is now a requirement in the revised version that the investigator must ensure that the person who's giving consent is the one who is signing it, in whom you are administering it. So Aadhaar is an option. Other countries have other identifiers. The whole point that you will have to take care of is the security of whatever document that you collect from the person. You'll have a copy of it. It has to be utmost security has to be maintained. Thank you.
Thank you.
So if there are no more questions, I can turn off my video, and thank you very much for your patient listening. Thank you.
Thank you.
Thank you, madam. Thank you very much.
Thank you, Shafiq madam, as well as Banale sir. Now we can go ahead with the next topic. And now I request Dr. Sarfraz sir to introduce second speaker, Dr. Santosh Taur sir, for the next session. This session will be for 30 minutes, followed by question and answer session for 10 minutes. Over to Sarfraz sir.
Yes, sir. Can you hear me, sir?
Go ahead, sir.
Yes, sir. Good afternoon, everybody. At the outset, I would like to thank Dr. Ganesh sir, Dr. Kamal sir for giving me the opportunity to be the moderator for this important session that is on handling protocol deviations and violation by Dr. Santosh Taur. And it is my honor to introduce the speaker to you. Dr. Santosh Taur is currently working as a Director of Medical Affairs, Pfizer India Limited. He has done his BM in clinical pharmacology, and he has a certified course in medicine development from IFAPP and King's College London. He has done postgraduate certificate program in digital health from IIM Raipur. He has more than 16 years of experience in clinical research, medical affairs, regulatory affairs, pharmacovigilance, and compliances. He is also a peer reviewer of two important journals like Journal of Postgraduate Medicine and Perspectives in Clinical Research. And he has almost 38 publications to his credit, including original articles and review articles. And he has also authored 2 chapters in a book, and he also has received several awards like OPPI Medical Excellence Award for Building Adult Immunization Ecosystem in 2025 and Champion of Health Award at the Indian Vaccine Leaders Conclave in 2024 and Hall of Fame Award by Indian Society for Clinical Research in 2021. So I feel privileged to invite Dr. Santosh Taur to deliver his talk on the topic handling protocol deviations and violations.
Thank you, Dr. Sarfraz, for this kind introduction, and good afternoon, everyone. I think we had wonderful talk on QBD, Quality by Design by Dr. Nusrat, and pleasure always to listen to her. What I will be doing now to give you a different perspective about protocol conduct. Okay, we all know in clinical trial, it is very important for us to abide by all the study related processes, documents, And this is not only from the compliance and the regulatory perspective, but also as a responsibility towards our research participants, because they have a lot of faith in us, the way we conduct the trial. They are contributing altruistically to the advancement of science, and that is it is all the more important for us to ensure that right, safety, and well-being of these participants is maintained throughout the conduct of the trial. And that is then this topic becomes so essential that we have to keep in mind the basic principles and learnings. See, what I'm going to show in my talk over next 25 minutes is that how do we define these protocol deviations and violations? I think the terminology has evolved, okay, and we need to keep pace with that. Uh, they were like almost a couple of decades where there was ambiguity around the definition, classification of deviations and violations. Now I think most of the regulatory authorities have come forward. They have attempted and successfully attempted to define and categorize the protocol deviations. And today's talk, I think, will try to throw light on a couple of these advancements. Uh, Before I start with, I just want to set the foundation of protocol compliance. My talk will evolve around three important guidance documents. So we have the regulatory authorities such as USFDA and EMA. Okay, this is the latest from the USFDA, which talks about protocol deviations for clinical investigators. Okay, so you can refer to this guidance document, almost a year-old document, but very beautifully captures the essence of protocol deviations, not only and how, but also when do we need to report. Okay, then we have ICH guidance document. I think we all are aware of ICH. It has released multiple guidance documents under the four domains: quality, efficacy, safety, okay, and multidisciplinary. Under efficacy, I think we have E3, which is the structure and content of clinical study reports. So in that, we have this R1, which is Q&A, question and answer, that clearly defines the protocol deviations and the way it needs to be reported. Yeah, both to the internal stakeholders as well as the external stakeholders. This is mainly from the sponsor's point of view. And then we have our own NDCT 2019 and multiple updates and revisions to NDCT rules, which also clearly defines what are protocol deviations and amendments and what are the reporting expectations. Okay, so those who are interested to read more, my today's talk is based on these three important guidance documents. So you can very well refer to these documents and get more understanding. Now, when we get into the definition part of it, okay, we know protocol deviations are any unapproved departure from the trial protocol or procedures, which is often unintentional. See, no one will do it intentionally. Okay, and I'll talk about some of those intentional deviations, when we do it and why it is permissible. Okay, but again, whether it is intentional or unintentional, it also means that we should acknowledge, identify, classify, and report. Okay, and that reason we should be aware what are the areas, there are weakest chains in the clinical trial conduct, which we realize over the period of time with experience, and then we have to have more focus, and that is today called as risk-based monitoring, because we have some areas which are identified as the high risk areas. Okay, then protocol violation. Protocol violation is a subcategory of deviation which imposes the serious non-compliance concerns. Okay, either regards to the data quality, completeness, accuracy of the data, or with regards to the participants, could be their safety, rights, or well-being. Okay, when we talk about rights, it also involves the informed consent form. Okay, the process of informed consent, safety, self-explanatory, and well-being. Like Hippocratic oath, we always say do no harm. Okay, so that's why protocol violations, they emerge. But over a period of time, it so happened that we had multiple definitions, like protocol deviations. In that, we had minor deviations and major deviations. Then we had critical protocol deviations. And some of these are also interchangeably called as protocol violations, because violation means something which is of a greater concern and that would have some action, either regulatory action or ethics action. Okay. I'll talk more about it, like how this terminology has evolved. But you keep hearing this terminology, so I thought let me introduce you to these concepts, and then you will understand what is the newer definition. Then there's a regulatory compliance importance, because both of these deviations as well as violations, they need to be documented and reported to maintain the transparency. And how does it impact? Definitely, because see, the clinical trial basis is patients' trust in us. We as clinical researchers, we as sponsors, we as ethics committee members, patients are entrusting us with their life. Okay. And that's the reason when the moment patient becomes a participant in the clinical trial, okay, the complete shift is towards the well-being, rights, and safety of these participants. Yeah. We all have to work with patient centricity because they are participants in your study. And then any of the deviations and violations, they would first breach the trust. Okay. Second, it will have impact on misclassification or underreporting, because sometimes these are all human tendencies. You tend to underreport any finding. Okay. And that's the reason we need to have very clear framework to ensure that this is these deviations and violations are not looked at as a negative feature. Because see, we are bound to have some deviations in a trial conduct. It will happen, but then it also important that we acknowledge and transparently report this deviation, and that's where the impact on the clinical trial comes from. Because if it is found at a later stage, at monitoring stage or audit or inspection, then it has completely different perception. That site may have implication in the future clinical trial selection. And that's the reason this is very important to understand the implications of these deviations on the c- ongoing trial as well as the future of the clinical trial. Just to give you some of these examples, like what are the protocol violations? So this was the article which was published almost a decade ago, which captured the commonly observed protocol violation. For example, informed consent process, the eligibility criteria are not met, and this will have safety implication. Unfortunately, if you recruit a participant who is supposed to be excluded because of either hepatic failure or renal failure, because we know the investigational medicinal product would have a pharmacokinetic profile which doesn't warrant the use of this medication in this risk group, and you inadvertently recruited that participant, so it imposes the safety risk to the participant, and that's the reason these violations, the eligibility criteria need to be adhered to strictly. Okay. Under reporting of serious adverse event, you cannot miss to report the adverse events, whether serious or otherwise. Okay. Blinded trials, improper breaking of the blind, it may not impose the safety risk, but what happens? It breaches the data integrity. Clear? Because it might introduce the bias. See why we are introducing blinding techniques in the clinical trials? Because we want completely reduced level of bias in the study. And if you have improper breaking, that introduces the bias and that may impact the data quality. Yeah. Missing test. If there are certain critical tests, for example, if you're doing a vaccine study and the approval of the vaccine is based on the immunobridging, because these days for different vaccines in the same category, we do not do efficacy trials unless it is a new vaccine. So what we do, for example, there is a pneumococcal vaccine, a valency vaccine has been approved, like 13 valency. Now you are working on a different valency, like 10 valency, 14 valency, or 20 valency. So what you do, you don't have to do the efficacy trial as per WHO and various health authorities. You do immunobridging. So what you do, you collect the immunogenicity samples from the participants and you compare the immunogenicity levels in the test group versus the standard of care, which is the existing vaccine. And based on the immunogenicity levels, you give the approval for the vaccine because there is no efficacy, because this is a bridging. You are showing that by the way of immunobridging, we are extrapolating the efficacy of the vaccine to the original, the first vaccine which is in this class. Now, if you miss to collect one of the immunogenicity endpoint, for example, 1 month after the primary season, infant vaccination, you have given 3 doses and you are supposed to collect the immunogenicity sample 1 month after the vaccination, after every dose. So you collected post dose 1, PD2, and post dose 3. And there's a fourth booster dose which is after 1 year, and you have to collect 1 month after that. If you miss to collect this sample, it becomes a protocol violation. Why? Because this is very important test to ascertain the immunobridging concept. Okay. And now if you miss to collect this, then it leads to protocol violation because it compromises the data completeness and the integrity. Okay. Sometimes we have given the window. For example, a patient is supposed to follow up at around 2 months. So what we generally do in the protocol, we give the window. The next follow up at 60 days plus minus either 7 days or 14 days, depending upon your protocol requirement. And this is permissible. So instead of 60 days, if patient comes for follow-up on 65th day, okay, this protocol allows to have that follow-up, you report it, you mention that, and you recruit that particular visit findings. Now, what if the patient comes on 79th day, which is outside the 7-day extra period? As per your protocol, you said that 60 days plus minus 7 days, which is permissible. So you can have follow-up as early as 53rd day, or you can have follow-up as late as 67th day. But anything beyond 67 days becomes protocol deviation. If it happens once, does not have any impact, it becomes minor protocol deviation. One time it happened. But imagine protocol deviations, especially the minor ones, which happens multiple times, then it may amount to your violation. Yeah. So you have to also remember it's not just the severity of the deviation, it is the frequency with which the deviations are happening. And that's the reason you see this point, multiple visit meets outside the permissible windows. See, single visit is still okay. You can report it, but still you have to report. But if it is happening multiple times, then that becomes a violation. Okay. Any intentional deviation from protocol, okay, which is as per the GCP or regulation, comes under the violation. And repeated non-compliance, very important. Now let's understand how does US FDA define. See, protocol deviation as per US FDA is any unintentional departure from the IRB, Institutional Review Board approved protocol. And protocol, we all know what does protocol include. Protocol includes the study conduct document, which is the core for study protocol, the monitoring plan, the statistical analysis plan, including your case record forms. Okay. All these things constitute your study protocol. So anything which is deviation from the requirement of this protocol becomes a deviation. Now, US FDA has categorized certain class of subset of deviation as important protocol deviation. So now these are not called violations. Okay, so they are deviation and important protocol deviation. So important protocol deviation is a subset which might significantly affect the completeness. You have missed certain important visit sample collection. Accuracy, error in reporting the test or the findings, or reliability of the study data, breach in the data log, breach in the blinding, yeah, or any deviation that might significantly affect the subject's rights, like informed consent form, UBS informed consent process, no AV recording for regulatory trial, participant signature missing, or forgery, safety, eligibility deviations, okay, or wrong investigational medicinal product is administered, or rescue medications not given when it was needed, or well-being. Okay, patient's well-being is compromised. So any deviation which would have impact on either of these becomes important protocol deviation. Yeah, and these important deviations, while they are these different terminologies, major, critical, significant, but FDA recommends using important to encompass all these terms. Okay, so that we do not get confused with the terminology. So they have become more simplistic. Just put everything under the bucket of important protocol deviation. Okay. Now let's look at our erstwhile definitions of minor and major, because in India we may still continue to follow some of these older definitions. Okay, because there are different sponsors. Every sponsor will follow ICH and the country of origin guideline framework in alignment with the Indian regulations and the ethical principles. Yeah. So that then it is also important to know what is minor or major. Yeah. So minor deviation, these are mostly administrative errors that do not affect the safety and well-being or the study validity, but they need to be reported. No matter whether it is minor or major, any deviation needs to be reported and documented. There is no hiding, because at later date, some other audit, trial, monitoring, inspection, this will surface. So it is always ethical and responsible behavior from the investigators to report the deviation irrespective of their severity and the frequency of this deviation. Okay. Major deviation, as we discussed, these are similar to what USFDA defines as the important protocol deviation, because they have significant impact on the data integrity or the participants' right, safety, and well-being. Okay. There are multiple ways of classifying it. Risk-based criteria. Depending upon what is the risk, I think sometimes these are classified as critical, major. Okay. What is important is that irrespective of the classification, why it is important to know, because these are the areas which will define our monitoring. I think we all know we have moved away from traditional monitoring in most of the trial to risk-based monitoring, because it is more resource efficient. And there are many examples which have shown that when you do risk-based monitoring in comparison to your traditional monitoring, whether it is source document verification, okay, uh, it has reduced the resources by almost 30%. The question is, does it pick up more finding? Yes. In fact, it has been shown to pick up more findings than the traditional, almost to the tune of 40 to 70% of it. So, so this is more efficient use of the resource. And that's the reason reporting these deviations gives better predictability and accuracy to your risk-based monitoring. Yeah. And now, important point is from the ethics committee perspective. Because see, how do you build trust in the conduct of your clinical trial? Yeah. It is through the transparent reporting. Why we report our clinical trial findings, whether they are positive or negative? It is mainly to ensure that regulatory and ethics committees, regulatory authorities and ethics committees, they have given us the permission to conduct the trial. We see the trial participants are putting their life in our hands, okay, by participating in this study. So it is our moral responsibility to report all the trial findings, whether they are favorable or unfavorable. And that's the reason it is very important for us to not only identify the protocol deviation, but to also do the root cause analysis, have the corrective plan, not only to manage the ongoing deviation, but also to prevent the future deviation. Yeah, we have to take learnings. And that's the reason this regulatory and ethics requirement is so important to prevent these deviations from happening. Otherwise, what becomes? It becomes a trend. We have seen certain sites, certain trials with repeated observation, and it becomes a systemic issue then. When you have certain observations repeatedly being reported from certain sites, that means there's something significant amiss there. and which calls for certain action, and we'll talk more about it. So this is our NDCT rule. I want to bring your attention to this aspect. See, we acknowledge that deviations are bound to happen, okay, and we have to report them. But also there are certain deviations which are acceptable to be implemented without prior written approval of the ethics committee or central licensing authority. And that is only when we have to eliminate immediate hazard to the trial subject. For example, we have done blinding, the trial is going on well. However, we known that there is a serious adverse event, and it is a life-threatening adverse event. We need to know which treatment arm does this patient belong to. And to manage that adverse event effectively, we have to know whether patient is in the investigational medicinal product arm or in the control arm. Control arm could be placebo or could be other standard of care. So when it becomes essential to break the blinding to save the life of the patient participant, the NDCT route permits us to do so. Remember, it has to be of that level. You cannot just use it under the garb of okay protecting lives. We have to report it. See, again, it still means that we need to report it, and it clearly mentions that all such exceptions must be immediately notified to the ethics committee as well as the CLA, Central Licensing Authority. But it defines the ones which are important protocol deviations, you have to report immediately, notify immediately. The ones which are other deviations, administrative, logistic changes, minor amendments in the protocol, which does not have any impact on the study conduct, data integrity, or participant safety, rights, and well-being. Those logistic administrative changes can be notified or should be notified to the health authority within 30 days. See here? So we need to follow this timeline very strictly. Let's talk about the deviation versus violation. I think we have spoken at length about it. What wish to bring focus here is more about why do we need to define them? Because our kappa will depend upon whether it is a deviation or important protocol deviation. We interchangeably call it violation. Now, let's look at what are the common deviations and causes. We have seen mostly they are around the enrollment criteria, informed consent issues, safety-related deviation, the missed lab test, adverse event reporting, safety monitoring, or the documentation. See, you have to remember the deviations, protocol deviations, and the finding, which are from the GCP perspective. See, there may be some GCP-related finding. For example, the signature, training signature missing. Is it the protocol requirement? No, but it is one of the GCP requirement. Okay. So we have to segregate the GCP related issues from the protocol deviation so that we do not overtly report everything. That's the reason protocol deviation is a sub bucket. GCP is very broad umbrella. Under that, there may be many deviation in reporting that we'll have to do. But what is important under protocol deviation is anything which is defined in the protocol. If you are deviating from that, that needs to be reported. And these are the common examples of protocol deviation. What are the root causes? Mainly the training and the workflow issues. There are certain communication and documentation gaps. What the communication between sponsor, investigators, and the study team members. I spoke about the significant organizational issues. We see certain sites having these recurrent issues, which shows the questions about the capability of the site staff, about the rigor and the oversight which is at the site. Okay. And also the root cause analysis of how we are approaching. See, if you have done the root cause analysis, you have to preventive action, then ideally the subsequent issues should be preventable. Yeah. And that is then something there are different ways. 5 whys, fishbone analysis, which are used for the root cause analysis. Like you identify the theme just like a fishbone, and you start deviating from the main theme and get detailed into the sub themes. Like, is this the consent issue or the safety issue or the visit issue? Then if it is a visit, then you start getting further. So that's called the fishbone analysis. So you have to follow this root cause method. Then you have to see what is the impact. You have to quantify the impact wherever possible. Okay. You have, when you report the issue, you not only report the root cause, you also report what is the potential or the existing impact of this deviation. Okay. Uh, are there any data integrity issues? Will it lead to delay in the study completion? Okay. There could be impact, like sponsor may get some warnings, but the investigational sites may have certain for cause audits happening. These are all the impacts of the protocol deviation. What are the expectations? See, ICMR guideline has clearly laid down the core principle, where topmost is the participants' safety, rights, and well-being, while respecting the autonomy and beneficial to no harm. So these guidelines also have provisions for the deviations management, reporting in timely manner to the ethics committee. It is important protocol deviations you have to immediately report because they raise important issues around the participant safety and the data integrity. Ethics committees have to enforce the corrective actions, sometimes suspend the enrollment, request protocol changes. If they observe there are certain deviations which are repeated, and the investigators they align with the sponsor and say that okay, we need to modify this eligibility criteria. The sponsor and the investigators should agree upon the protocol amendment, and that ethics committee has to approve. Regulatory expectations, I just showed you the NTCT rule, which clearly calls out that no matter what is the severity of the protocol deviation, you have to report it. Some may be reported immediately because those are important. Others, minor administrative, can report within 30 days. But it is very important from the QC point of view, quality management, that you have strict reporting timelines. Okay, we discussed about it. It is everyone's responsibility. Investigator has to immediately report to sponsor and the ethics committees. Sponsor has to report it to multiple authorities, especially in case of multi-country, multi-centric clinical trial. And ethics committee has to take the timely action. Yeah. And I showed you this more from the timeline perspective, 30 days for minor administrative logistic changes and immediately for all other important protocol deviations. And this is the ICH E3 R1 framework, which clearly shows in the clinical study report how you are supposed to mention the protocol deviations. Okay, so this is the framework template. You may have algorithm, and this is how you report the protocol deviations. Now, talking about the corrective action. Corrective action, it addresses the immediate cause, but preventive action, it prevents, eliminate the root cause to prevent the recurrence of this. And that's the reason this CAPA has to be aligned both with the HS committee and sometimes with the health authorities also. When there is an inspection and the inspection demands you to provide CAPA, then you need to really give a detailed CAPA plan. Yeah. These are some of the disease inspection findings, which is available as an anonymous report. Okay. We need to identify the common finding, and we as sponsors and investigators and ethics committees should be aware about these issues so that we can tighten our vigilance around these issues. Systemic quality issues, repeated observation, quality management evaluation, training related, oversight related issues, proactive compliance alignment, reporting these issues proactively, and not to be found out at the time of inspection or audits. So to summarize, we have the best practice of protocol training, spending more time in training of the study staff, clear delegation and version control, so that there is a communication between sponsor, investigators, and the study team about what are the protocol related requirements and the timeline. Risk-based monitoring plays a critical role to identify both reported as well as unreported protocol deviations. And this is very important. We still see there are many unreported protocol deviations which surface during the monitoring. That's the reason monitoring plays very important. And then there should be non-punitive reporting. And that's where the role of the investigator sites comes into picture. How you build that culture of trust. It's not the culture of punishment that you find something, you have the punishment for the concerned staff. You have to build a culture of trust where they feel empowered to report any deviation. And that will help in the long term to get more trials to the set. To summarize, we need to know first of all what are protocol deviation and how to differentiate them from other deviations from the GCP perspective. And you do not over-report everything. Prioritizing ethics and safety at every stage of the trial conduct. Focus should be on the quality and having the robust QC/QA plan, building the compliance culture. And this is the ultimate motivator to have very robust clinical trial framework which will ensure the safety, rights, and well-being of the participants. With this, I'm opening this forum for any questions and answers.
Thank you, Dr. Santosh, for your excellent presentation, insightful presentation on the topic handling protocol deviations and violations. And I think the floor is open for question and answer session. I can see one question here from Arjun sir. What are accepted protocol deviations?
Sir,
Unmute,
sir. Sir.
Unmute, sir. Santosh, sir.
Yeah. Okay. So if you ask from the sponsor, investigator, regulators, and ethics perspective, I will not define what is acceptable. Frankly speaking, quality by design, we just heard, is given. We have to have the highest standard of quality where we have zero deviation, zero violation. Okay? What I'm saying is not what is acceptable. It is what is minor and what is important. That is something that we should always strive for zero deviation. See, you cannot have acceptable eligibility criteria deviation. No, I cannot have. You cannot have acceptable study visit deviation. So whatever is acceptable becomes part of your protocol, to be very candid. And that's the reason, like I give an example of window, sampling window. Like for we have given 60 plus minus 7 days. Now, and this is the importance of having the site initiation. And before that also, the protocol, visits and alignment, where we discuss with the investigators on ground. If we have 60 days window, what is your practical experience? See, because we have to see the practical side of the patient's follow-up as well as the requirement. Why we have follow-up window of 3 days for certain, 7 days and 14 days. Because there are certain follow-ups where if you have follow-up later than 7 days, it will change the pathology. It will change the baseline parameter. It will change the root cause analysis or it will change the causative pathway. That's the reason I think we need to have very clear. See, this is not whimsical. I want 60 plus 14, so I'll keep 60 plus 14. No. So there is a science and the practicality. They come together when we define such type of acceptable window period. And that's where I would say whatever is permissible, like HP events. I'll give you an example. Like most of the times we see there are certain rare disease trials where we have given the upper age cutoff. Unfortunately, we realized that there is one patient who is like 1 week older than the upper cutoff. Then investigator is in a dilemma. Shall I recruit, not recruit? Because this is a rare disease. Anyways, I will not get this patient again. So in such exceptional scenario, with the prior permission from the sponsor, from the ethics committee, and if it is a regulatory trial, from the concerned regulatory authority, you can still go ahead and recruit that participant because lay down your rationale why you are going ahead and recruiting that participant. And in future, if you see that this is a recurring phenomenon, initially you thought that the rare disease is only between the age of 18 to 45. But in India, we see more older patients presenting with this condition. Like for example, cardiomyopathy. We thought that it is mainly in the middle age, but then we see it in the younger age group. Do we need to lower the age group or extend the age group? So that's where the acceptability comes into picture. So and that should get into the protocol and not as as an unsaid rule. We should not work with that acceptability in our mind when we are conducting trial.
Thank you. Another question is there, are there any timeline for reporting major violations like SAE, like serious adverse event?
Yeah, so the SA is not a deviation. So let me first clarify. SA is your reporting responsibility. Any SA, you have to report it as per the timeline. Like sponsor 24-hour timeline is there, EC timelines are there. So you have to follow that. What is, see, the timelines which were mentioned, they were for the protocol deviations. SAs are not protocol deviation. What is protocol deviation? Non-reporting of SA is a protocol deviation. So if you ask me the timeline for non-reporting, you have to immediately report because this is an important protocol deviation. And non-reporting of SA becomes a protocol violation,
Actually,
whatever definition we follow.
I think
Yeah?
the questioner wants to know, is there any timeline for reporting major deviations? Okay.
Yes, major deviations immediately.
Immediately. Okay.
And that's what I think NDCT rule 2019 also clearly says. You have to immediately report it as soon as you become aware of the deviation.
Okay.
Yeah. Minor one within 30 days.
Within 30. Okay. There's another question. Can repeated minor deviations be termed as violations?
Yes. And I explained that some time back. Because it is a systemic issue then. Yeah. And you have to then label it as protocol, important protocol deviation, and the corrective and preventive action needs to be taken because it exposes the limitations of the trial site. That either there is a technique gap, or there is understanding gap, or there is a culture gap. And you cannot put the safety of the patients, participants at risk. You cannot put data integrity at the compromise. Yeah, that then becomes a major deviation. Repeated.
Okay.
Kind of
Thank you, Dr. Santosh, for your kind or for your responses. And I hand over the session to Dr. Ganesh sir for further proceeding.
Thank you to both of you. Now we are toward the end of our session. We are very thankful to both the speakers, Dr. Shafiq Madam as well as Dr. Taur sir, for their excellent presentation, as well as moderators, Dr. Bhannale sir and Dr. Sarfaraz sir for their nice moderation. Lastly, my sincere thanks to all participants for their active participation throughout the workshop. In the last, my special thanks goes to Dr. Kamal sir, who was instrumental for success of this workshop. Sir has put their wholeheartedly efforts for this workshop and played a very active role and shown deep involvement since the inception of this workshop. So please give a big round of applause for the sir. And now I request Dr. Kamal sir to address the gathering for 2 to 3 minutes.
Thank you very much, sir. Thank you very much, sir, Dakle sir. And congratulations to all the speakers. Both today's speakers have been fabulous. Dr. Santosh Tor and Madam Nusrat Shafiq, and both of the supportive moderator, Dr. Sarfraz and Dr. Shashidhar Banale. We pay sincere gratitude from National Association of Pharmacology and Therapeutics. And with this ending this session, again, Dr. Dakle's hard work, who is heading the NPT at present, he has become the country's largest organizer of the GCP workshop. And probably today's, I can say with confidence, he has trained more than 5,000 doctors to GCP training. A big round of applause to Dr. Ganesh Dakle sir and the entire team for leading from the front. Thank you to all the participants who have supported us very gracefully and attended all this session thoroughly of this National GCP workshop. We have made a certain arrangement for the easy access to all the sessions by all the participants. The video recording and the resource material have been regularly been updated of the past all the sessions, and today's session will be uploaded by the evening of today. We sincerely thanks to all the participants for supporting this initiative, national initiative by National Association of Pharmacology and Therapeutics. Your certificates can be downloaded within the period of 7 days from today. and we are behind the scene working day and night. You can access to your dashboard. Your WhatsApp number is your ID, and you can always change your password for accessing your dashboard. So please ensure that you have the access of the dashboard, because the dashboard is one point access for all the related queries that is related to the GCP workshop. Thank you again for making this a grand success. Was our 5th edition under the able leadership of Dr. Ganesh Dhakle, and all the speakers and support persons and resource persons have been done a fabulous job for completing this task. Thank you to all. Long live NPT. Jai Hind.
Thank you, Dr. Kamal sir, for your address. As mentioned by Dr. Kamal sir, certificate will be given to our participant very shortly. So once again, thank you all to make this online workshop grand success and memorable one. With this, I declare that this workshop of 4 session is over now. Thank you to all. Thank you, madam.
Thank you very
Thank
much.
you,
Thank you,
Santosh.
sir.
All the
Thank
best.
you,
Thank you, sir.
Sarfaraj, as well as Bhanale, sir.
Thank you, sir.
Thank
Thank you,
you.
sir.
Bye-bye.
Shilpa, you can end the session. Thank you.
Okay, sir.